In the previous couple of videos, we saw how cells differentiate themselves. They use cell-to-cell communication to determine which part of the body they are in, and then once they receive signals from other cells, they start making RNA molecules from their genes, from the DNA that they have. They start making only those RNA molecules associated with the correct cell type. So if you realize you're a skin cell, you only make the genes. You only use the genes that are associated with skin cells. If you are a stomach cell, you only make the cells that are associated with being a stomach cell. Easy enough. We also saw that the way that in a fruit fly and other organisms, we determine which side of the embryo is going to be the head and which side is going to be the tail, where the arm's going to be, the leg's going to be, is by making all of these short segments of RNA that are indicative of this is the head, this is the tail, right? Only cells with that kind of gene active will be part of the head or part of the tail. Now, if you think about it, any gene that the cell is using will have some RNA floating around the cell associated with it. Because to use a gene, you have to transcribe it, make some RNA, and then translate that RNA. So any gene that's active in a given cell type will have some little bits of mRNA floating around inside the cell. Sometimes scientists have cataloged the entire genome for a certain species, but we don't know which genes are active in each cell type. We need to figure that part out in another step. So what we do is something we call nucleic acid hybridization. So let's say I have a section of mRNA for gene A floating around, right? Gene A. So this was made from the DNA template. I want to know all of the cells that use gene A. So what I'm going to do is make a molecule of DNA that matches up with that mRNA. Now you might think, well, why don't you just take the DNA from the genome itself? Why are you bothering to take the RNA and make DNA? Clearly you have the organism, why don't you just grab the DNA from there? But remember, a DNA molecule has thousands and thousands of genes on it. I'm only interested in one gene right now. So I'm going to use this trick and take the mRNA for that one gene and make just a short section of DNA that is complementary with it, that will attach to that mRNA if it's present. And that's called cDNA, complementary DNA. We do this with an enzyme called reverse transcriptase. Now, we're going to take our cDNA, we're going to attach it to a glass plate. This is the cDNA for gene A, B, C, and D. To each of these complementary DNA sections, I'm going to attach a fluorescent tag. This will show up under a black light. It'll glow under a black light. So, I can do this for every single gene inside that genome. I can make a short section of DNA that will match up with the mRNA, and the mRNA will only show up in the cells where that DNA is being used. So, let's say I take a certain cell type, and that cell type only uses genes A and gene C. I can take a cell of that type, I can blend it up so I get a nice cellular slurry. I get all that cytoplasm with the mRNA mixed in. And then I can spread that mRNA over all of these cDNAs, all these little sections of DNA that are attached to that glass plate. Gene A, the mRNA for gene A will attach to the cDNA for A. Gene C, the mRNA will attach to this one. And once those attach, what will happen is those sections will start glowing. So I have a plate, right? I have this whole plate here. And I've put sections of cDNA in all these little divots on the plate. And each one of these represents one gene inside the genome. So, when I spread all the cytoplasm from one cell type over this plate, what happens is I get something like this. Only the genes that have mRNA in that cytoplasm, that is, the genes that are actually being used by a cell type, will glow on this plate. So I can figure out every single gene that's being used by a cell type by using this system. This is called nucleic acid hybridization. And this glass plate here with all the different little sections of DNA attached to it is called a DNA microarray. So this was a complex bit of information because there's a lot of moving parts here. To review, each cell type only uses some genes and not others. If it's using a gene, it will make mRNA for that gene. If we set it up so that every gene section, every segment of DNA for a gene, is arranged on this DNA microarray, we've attached it to the glass, and it has a tag which fluoresces when the mRNA for that gene is present, then we can take any cell type, spread its contents over this plate, and only the genes with active mRNA will actually glow. So I can figure out which genes are active in skin cells versus which genes are active in bone cells. And I can do this for any species. So we can figure out which parts of the genome are relevant for leaves versus stems, or roots versus shoots. We can do it for a sea sponge and see if there's any difference between cells that are on the inside of the sea sponge versus the outside of the sea sponge. This is just a way for us to visualize which genes are active inside of the cell. So, researchers can monitor the expression of specific genes. Scientists use various techniques to study how genes work together, one of which is nucleic acid hybridization, which allows researchers to identify cells in which a target gene is expressed. A DNA microarray can gather data about which genes are turned on and off in a particular cell. Here's how it works, right? We get a section of mRNA and a molecule, an enzyme called reverse transcriptase. Remember, transcription is going from DNA to RNA. So reverse transcriptase takes RNA and it makes complementary DNA. It takes that mRNA molecule and it makes a short section of DNA that basically is the gene, just one little section of DNA that's just the gene that we're interested in. We then add a fluorescent tag to each of those little gene segments, and then we find out which cells are using that gene or which cells are not using that gene. Then we can take information from every single cell that we've gotten these results for, and we can start to put together a diagram of which genes are active in which cell type. So this is a fruit fly larvae, like the ones we saw in the previous video, but it shows, oh, these genes are only active in this strip, nervous system, the basic nerve cord going down the back here, that are in the center. These genes are only active in these horizontal slats that represent each body segment going through. You can see different genes active in different locations. So this is one of the ways that we know which kinds of genes are active in each tissue type. We can actually sample several genes at the same time if we're a little bit clever about it. You see there's different types of glowing proteins that we can add in here. So we could have gene A glow red in these little dots on the glass plate, and gene B glows green. So anytime you see a green one, that means that gene B is active, but not gene A. Anytime you see a red dot, like this one over here, that means that gene A is active, but not gene B. But when red and green light combine together, it expresses yellow light. The way paint mixes in pigments is not the same way that colors of light combine when you mix them together. So green and red will make bright yellow light as a result. And anything that's dark is not expressing any of these genes. So we have gene A represented by the red wells, gene B represented by the green wells, both gene A and gene B represented by the yellow wells, and then the dark wells are ones where neither of those genes are being activated.